Okay, hello everybody. Thanks for staying all the way to the end. I'll try to be fast so we can go to Jackson environment. So, well, my name is Sebastian and I work with two advisors. One of them is an expert in optogenetics, the other one is a computational modeler, and I'm trying to apply these two techniques to characterize CRISPR dynamics. So, let me begin by describing very briefly what CRISPR is. This is a variation of this system CRISPR that a lot of people have been using in the last couple of years to edit for genome editing and for some other purposes. CRISPR is a variation that allows for transcriptional repression. And basically it's composed of two elements, a protein called Cas9, and which is a catalytic inactive nuclease, and a piece of non-coding RNA called single guide RNA, or sgRNA for short. This sgRNA is composed of two parts, the base pair region and the Cas9 handle. The Cas9 handle is what allows it to bind to Cas9 and form a complex, and then the base pair region allows this complex to be directed to some part of the DNA. And if this part of the DNA happens to be the coding sequence of a gene or a promoter of a gene, it will block transcription and it will effectively act as a transcriptional repressor. This is interesting for people trying to build synthetic genetic circuits because you can easily see that by varying the base pair region, you can get practically unlimited number of repressors. And this is nice because people in trying to build synthetic genetic circuits have been limited for approximately 10 years by the number of parts that they have had to build logic gates, oscillators, and other kinds of circuits. But CRISPR solves this problem, and some researchers have already taken advantage of this fact to build the simplest type of circuits that you can build, which are logic digital circuits. So for example, if I can control the expression of one single RNA with one promoter, I can have an inverter or a NOT gate. Because if I activate transcription from this promoter, I will repress a second promoter, which is the output promoter. So for an input of 1, I will have an output of 0, and vice versa. And you can take this analogy further and make other kinds of logic gates and make more complicated circuits. And in fact, this has been done, this has been presented in a paper at the end of last year, and people are still trying to do that in higher organisms in eukaryotes or yeast. What about dynamic circuits, circuits in which dynamics actually matter? This hasn't been reported yet, and the reason is that characterization of dynamics of gene circuits in general, or gene elements, has been a lot more challenging. There are a bunch of issues that I will talk about in the next couple of slides. But if this is overcome, this is done, then we will have a lot more circuit elements to play with in synthetic genetic circuits, such as timers, toggle switches, or oscillators. And I'm talking about big number of these elements in the same cell all interacting with one another, as opposed to what has previously been reported, which is one of these at a time in a single cell. And this, of course, requires a very thorough characterization of CRISPR dynamics, which is basically what I'm trying to do. So we basically in my lab try to characterize dynamics of different genetic networks. We do this by taking this analogy from electrical engineering, in which we have a device called function generator, an electronic function generator that we connect to an electronic device that we want to characterize. And we perturb this device with different kinds of waveforms. We read the output, and by that we can build mathematical models of this circuit element. And the same idea can be carried out to biology, and we can, and in fact we published this last year, how we can build a biological function generator in E. coli that is activated by light, and how using light we can feed arbitrary signals to circuit elements and characterize those circuit elements better and build dynamic models of them. So I'm going to briefly describe the function generator. This is what's called a two-component system. This is done in E. coli, and this is a two-component system that is sensitive to light. So just to make it brief, under green light, this two-component system controls transcription, activates transcription from an output promoter called CTC2, and then under red light, transcription from this promoter is deactivated. We can regulate this under a wide variety of intensities of green light, and we can get different levels of expression from the output reporter. And furthermore, we can look at dynamics. We can look at how activation from this promoter goes up in time when we switch from red light to green light. We call this deactivation dynamics. And that deactivation dynamics is the opposite, is when we switch from green light to red light. And we can see how expression from the output promoter decreases, which makes sense. And the only thing that I want to highlight from this is that these two responses are very symmetrical. They can be fit by a standard second-order linear system. Both responses to the same system with a time constant, half-time constant of around 2 subdivisions. So now we want to actually characterize a single
single RNA inverter that we talked about. So we have considered the expression of the Cas9, and we connect the light switchable two-component system or function generator to an scRNA, and we are basically going to control transcription of this scRNA. These two elements are going to combine and repress transcription from an output promoter that expresses GFP. So the first obvious thing is to just look at the steady state response of scRNA inverter, and we can look at how expression of the output reporter varies as a function of the scRNA, and it goes down as it's expected. One thing to point out from this response is that it is non-sigmoidal as opposed to other transcription factors that have been reported like LAC-I or TEGAR. And the expression decays sharply, and it stays at the minimum value pretty much with low levels of scRNA. So dynamics is a little more interesting. Let's look at the repression dynamics. What happens when I switch from minimum expression of scRNA to maximum expression of scRNA? And of course, the output reporting goes down in time, which makes sense. You can also look at the opposite. What happens if I switch from maximum production of scRNA to minimum production? And of course, the response is going to go up in time. And what's interesting here, though, is that these two are very asymmetrical. Repression happens very fast within approximately 1.4 cell divisions, whereas the repression happens a lot more slowly, and it has this sort of this pure delay stage, and then what follows can be very well approximated by a second order stage. So there's this asymmetry, and we thought this was interesting. So in order to explain or to get a sense of why this could be happening, we developed a mathematical model of gene circuit based on CRISPR-I. So we have considered the expression of the Cas9, controllable expression of an scRNA that is actively degraded in the cell. These two things combine into a complex. There's an output promoter with an output reporter, and the Cas9 scRNA complex combined to the promoter and, of course, inactivated. So this results in a reasonably simple 5-equation model. This is an ODE model, a simple ordinary differential equation model with 5 species, as I just described. And just as a first pass, I took parameters from the literature from in vitro studies of CRISPR-I. And I just wanted to see with these parameters, without explicit fitting, how well these would be able to fit the experimental results. And it turns out that they can do it reasonably well. So if I look at the states of response again, it goes down, which makes sense, but it's non-sigmoidal as I observed in the experiments. And then if I look at dynamics, I also observe the same asymmetry. Repression goes down very quickly, whereas the repression also has this fixed delay stage at the beginning, and then a second order increase in expression following that. So this is kind of cool. The next thing that I try to do is try to do explicit fitting of the parameters of the model to the experimental response. And just to get a sense of what I'm supposed to get here, it turns out that there are several kinds of scRNAs depending on where they target in the promoter. And people have studied how these things affect, how the specific place in the promoter affects the amount of repression that you can get from the system. And I look at the design of the scRNA that I have, and it turns out that it's not the best scRNA in terms of repression, but it's interesting in terms of dynamics, because when I fit the model, when I fit the parameters to the experimental response, I get at least 2 parameters that are very different from the in vitro parameters, which are unbinding constant of the scRNA Cas9 complex from the promoter, and then there needs to be expression from the repressor promoter. We're looking into different kinds of scRNAs to see if we can parameterize all these scRNAs and make basically a data sheet of all these inverters and understand how they vary in dynamics and make a specification of each one of these. So I just want to conclude highlighting how I think I could achieve these 3 elements that I mentioned at the beginning: timers, toggle switches, and oscillators. So timers is very interesting because I'm hoping that I can essentially program how much I can wait until some gene is expressed. And to see how that can be achieved, I want to look back at the repression dynamics. As I said, there's like a pure delay, stage, and then there's a second order production stage. And why is this happening? At least the model gives us some insight into this. And the explanation is that if I am expressing some amount of scRNA before time 0, and then at time 0 I stop production of scRNA completely, I'm still going to have some amount of Cas9 scRNA floating in the cell. And I'm assuming that this is stable, as many other proteins in bacteria. but this still needs to decrease because of cell dilution, because of cell growth. And this needs to go below the concentration of my promoter. And once that happens, only once that happens, I will be able to produce new copies of the promoter that will not be repressed, and therefore I'll have active promoter which will express active output recorder. It follows from this that I can do the previous repression step with different amounts of sRNAs, and I can play with the pure delay stage right here. And if I do those simulations, it turns out that I can go as far as I want in terms of the fixed delay at the beginning. So very quickly, it turns out that a total switch can be achieved even though the response is not sigmoidal, at least according to the simulations. So I can do a phase diagram of two sRNAs mutually repressing one another, and it turns out that the system is bistable. I'm still looking into what are the conditions that guarantee this behavior to be true. It might be that the copy number of promoter plays a role, and I'm still not sure. I'm still working on this. And last thing, similarly, the oscillator, I add an additional sRNA to the loop. And it turns out that the system can oscillate, and if I, again, do a phase diagram of total sRNA 1 versus 2 versus 3, I can see an cycle. Again, I'm still investigating what will guarantee this to happen in the cell, and how I can play with, for example, promoter strengths to get a tunable period or tunable output. And with that, I want to thank my two advisors, Jeff Tabor, Oleg Goshin, and their groups. They've been really helpful in this project. And funding, which is coming from the NSF, and of course, RISE and the CCP and the organizers of this conference. Thank you.
Time for questions?
In addition to playing with the level of sgRNA, would it be reasonable to destabilize the sgRNA somehow through degradation or changing the sequence in some way to achieve a faster speed for the drone?
I believe the sgRNA is, okay, there are two parts, or three answers to that question actually. I believe the sgRNA is reasonably unstable at this point, except when it binds to the Cas9. So the sgRNA is, at least with the system that currently has the standard mRNA, is going to be actively pretty fast. Then the second part would be, I guess there are only two parts. The other thing that you would think of is making the Cas9 unstable. So it's actually created by the proteolytic machinery, if we call it. But it turns out it's such a big protein that if we try this experimentally, the cells grow horribly, and such a good response is messed up. So, I mean, it makes sense probably theoretically, but once you put it in the cell, it just doesn't work.
I don't know if you know it not, but experimentally it's very difficult to get the CRISPR technology to work reproducibly, and I think a lot of it has to do with the geometric and temporal proximity to Cas9 and the guide RNA. And I don't know whether you've thought about it. It seems like your kinetic model might offer insight into why there's such huge variability experimentally.
I haven't seen that. For all the results that I have, pretty much show that CRISPR is the most reproducible, well-behaved system that compared to other transcription factors. At least that's my experience.
Including in the model. Have you done it experimentally?
I've shown experimental results. Yeah, all these thoughts that I had at the beginning, those are experimental results.
Those are all CRISPRized.
Yes.
Well, bless you.